Hello, everyone, and welcome to today's Indoor Air Quality Matters online workshop, How Bipolar Ionization Fits into a Healthy Building Plan. I'm Troy Raszka. I'm the Director of Marketing here at Pure Air Control Services, and with me today is our special guest, Dr. Rajiv Sahay, who is our Chief Scientific Officer and the Director of our Environmental Diagnostics Laboratory.
Hello and welcome to all the webinar participants, and thank you, Troy, for inviting me for today's session.
Yeah, so today we're here to talk about a recent study that was peer-reviewed and published by the Continental Automated Buildings Association, and that study was conducted here at our Environmental Diagnostics Laboratory and was headed up and written primarily by Dr. Sahay. So that is what today's webinar is going to cover, is our study of bipolar ionization that was conducted during the pandemic. So let's get started. So today we're going to just look at these few topics, and we're going to take you through not only what the white paper was about, what the study was about, but how we conducted the study, and then what our results and findings were of that study. So as such, we're going to take a look at the indoor environment and how that's defined, what kind of contamination sources can exist within the indoor environment, how to maintain or enhance acceptable indoor air quality, and then we're going to get right into the study, which is what is bipolar ionization, how the study was constructed, and then how it was executed and what the findings were. Of course, there will be time at the end for questions and answers, so that we ask that you just go ahead and type all of your questions into the question tray here in the GoTo platform. And also, I'd like to make a note that we do have some handouts available for download out of the handouts tray, and that is an actual copy, a PDF copy of this white paper, as well as this slide deck, and a little bit of background information on our environmental test chamber, which some of this study was conducted in. So Dr. Sahay, I think before we even understand this air cleaning technology that is bipolar ionization, we must first sort of take a look at the indoor environment and define what that indoor environment or indoor air quality is. So why don't you just sort of briefly take us through how the lab defines the indoor environment?
Very good. I'm pretty sure that before you dive deep in this subject matter, you need to understand that what we are doing. So very first word is indoor environment, and I'm pretty sure like many of you have various definition of indoor environment, and people practices various approach to define indoor environment. But in nutshell, we can define that anything in our surrounding and their relationship. So if we are indoors, meaning if we are limited by a boundary of something like a wall or cave or building or whatever, that is like indoor because it is separated from the outer atmosphere. So the moment you separate yourself in a confined environment, that can be like, a size of office, can be your whole building or anything which is separating you from the exposed environment, that is first of all indoor environment. Now, why this topic become very important? Because we share this space, the indoor environment. When I just explain what it is, there is a space, right? So that space, sometimes you share with others, sometimes you do not share with others, you are just have by yourself in that. But at the same time, there is a number of factors continuously interacting on your dwelling or your occupancy. So we have to be making sure that those factors, which is completely like, in surrounding environment, how they are going to influence you in a good way or in a bad way, right? So that is a total quality approach to assess that how good is good, how bad is bad. Nobody has defined exactly in terms, and there is a lot of work is needed in order to just make sure that kind of delineation happen. But I'm not going, today's topic is not about the indoor environment, so I leave that thing just there. Just consider the indoor environment is a confined environment which is separated from the outdoor environment or outdoor natural environment. So if you understand the indoor environment, why we are worried about the indoor environment and air quality. So there are two terms here. Now the environment, I explain you, then there is a subset, because environment encompasses or include a number of things, including air, because as you know that human being cannot go without air. So air is a very important component of the whole equation. So what kind of air you are breathing, or what kind of air you are living off? So that is the air quality. So that is another topic. So today's goal to find out that how we optimize or what bipolar ionization or other air cleaning technology is going to do in order to make those environment optimal or quality, or you can say acceptable. So before I go any further, why it is important to understand, and our viewer, many of them I can see that they are building managers, building maintenance people, OEM people. But first thing we need to understand why we are going to do the quality assessment and where the factors are coming that is making you think even about this topic. So the first thing is building type. It depends like what kind of building you're talking about, so that you can identify your opportunities and limitations. For an example, the building can be the hospital building, which we can be discussing more in our preceding slides. It can be a residential building. It can be an industrial setup. It can be a non-industrial setup. Building function. Many of you, except those who are directly handling the building situation, taking these things seriously. But when we say building function, what building function meaning? Meaning that the building is designed in a certain way that it can provide you a good occupancy. So the building function is a total sum of whatever designing is being done, including the air conveyance system, because most of the time due to the energy conservation we have closed system, meaning that we do not have that much of open ventilation. So we have controlled the ventilation utilizing the mechanical means. So that is talks about the building function. So the building function varies depending upon how it is being designed. Third and very important and crucial factor, which I say out of sight, out of mind. Each day we see this activity. Construction, renovation. You might have seen a lot of contractors putting polythene sheet across where the construction is happening. Many times they sprinkle water. Why they are doing it? Because when you do this kind of activity, then a number of particulates matter, both abiological or biological in origin, or various kind of chemical, are going to be set off in the environment. So where it is going to go? It is going to go in the open environment, and then again it is going to recycle back in our occupiable space by various means, which we will be thinking in our next slide. So why we are worried about all these? Because all these factors which I just explained you a minute ago directly or have potential to impact your health and hygiene. When I talk to health, meaning health of the building itself or health to the occupants, and hygiene, because if, and again there is a subjectivity there which I will clarify in some few next slide, that if you are not in a hygienic place, how it helps to proliferate the pathogens, allergens, which have a direct consequence on your health, or you can say it adversely affect your health. Make sense? Now, if that is the case, then the productivity, if either we are in home or in an office setup, if you feel lethargic, you're not that active, then it is total loss of productivity, not only in terms of money, but in terms of your health. But I quickly want to give you an example that the cost of indoor environment is not that big than the cost of an occupant there. If you want me to give a rationale on that, like if your employee or if the occupant's worth like $200 then per hour, then the indoor air quality might be like only $20. So if you want to have $200 save, or you are going to have $20 save. So these are some factors in the world of production make me people think that how we can optimize our environment so we are going to do some good for the society, being proactive, being productive. These are the words is being used. Make sense, Troy?
It makes perfect sense. So in other words, the type of building, what it's being used for, the age of the building, is there anything going on in the building that's not typical of normal use? All of these things can affect the kinds of contamination that can come into the occupiable space. Is that correct, Dr. Sahay?
Yes, and that's why this particular slide can give you a pictorial representation that what we mean when we talk about contamination. Again, contamination is a word frequently used that anything unwanted in your occupiable space that can also be visualized as contamination because you don't want them, but they are going to be there. So they are contamination. So the whole contamination can be divided into two parts depending on their origin. Some of them are having no biological origin, nothing has to do with biology. What are those? Such as particle matters. You might have heard PM10, PM2.5, PM5, etc. So those are very, very small particles which can go and impact your upper or lower respiratory organs because we can breathe them, but there is no medical significance there because they are not originated by any living source. This might come from the stone cutting, that might come from the dust, that might come from any debris or any kind of other particulate like rusting, etc., corrosion, that kind of thing may be the source of these particles. Not only that, but if you look on the higher level, then as I said you, the particles which is not generated from the biology is typically understand in that. But I will tell you some of the biological particles are also qualified in that range, but delineation is that they are biologically originated. And we have a way to determine whether it is biologically originated or abiologically originated. All kind of chemicals, volatile organic compounds, varied kind of phosphorus, minerals, etc. that can also be listed as a biological contaminant. Gases, carbon dioxide, carbon monoxide, etc. Various kind of fibers such as fiberglass, withering of your shirt, your sofa seat, your upholsteries, that is going to end up giving you fibers. So that is the whole another category which we study is there here at ED Lab to identify them, to categorize them, to quantify them. This kind of a study we do. And of course, solid waste, you see all kind of recycling of solid waste. Why we are emphasizing on that? Because solid waste itself, a big source of a biological contaminant. Having said that, there are certain contaminants that must have a biological origin of source, such as microbiological organisms, such as virus. Everybody is knowing SARS nowadays. It's a very common word. COVID-19. These are the words. Do you know that this kind of infection happen due to exposure or upon exposure of a virus called as coronavirus? So bacteria, mold, protozoa, insect scale. These all are biologically originated particle, pollen grains. And other whole category is not a live organism itself, but some point of time it was part of a live organism or originated from there, such as plant trichomes. This is a hair, but it is like, growing aerial part of the plant. And then due to the trampling or other mechanical means or by strong wind and things like that, they kind of detach from its original parents, meaning the plant, and come in air, and then we can go and have sometime the exposure, because we are going to expose around that, because these things come in our environment. That makes sense? So in nutshell, the contaminants are two types, a biological and biological. And here I can show you some of the picture that this is some kind of like, biological source, which is like, various kind of fungus. So they are there, they may be pathogenic in nature. If they are in pathogenic nature, you have trouble. These are some good example of pollen grains that is directly coming from the plant. And these are the example of non-biological or living thing that can be contaminant, such as opaque particle, brown particle, etc. Some hair you can see, or some man-made fiber. These are the abiologically originated particles, and that should be also considered when we are talking about the indoor environment.
So, Dr. Sahay, these types of contaminants can be both airborne and can be picked up off of surfaces. Is that correct?
Yes, that can be airborne, that can be surface borne, but do not challenge me with a question that chicken come first or egg come first, right? So both are like, there is a theory that like, if you have a source, then you have the aerobiological particles, air particulate. And if the air particulates are there, then some point of time they are going to settle on the surface, and then they can be regenerated from there. So I do not know the answer whether chicken come first or egg come first, but I think that both exist. And that's what I want to respond against that question.
Exactly. And then, of course, there's some other types of contamination that would be considered environmental contamination that could affect the productivity or the health of an occupant. Is that right, Dr. Sahay?
Yes, and that's why I put the another whole category, which is like, sometime overlooked by many evaluators, but it has a very, very impactful role in the whole indoor air contamination. So some of them, there are a number of others, but due to paucity of time, I'm going to take only 4 of these particles, which I wanted to explain or elaborate, because that has direct impact. The very first one is the temperature. Temperature has a very good impact on both biological as well as abiological particles. As many of you know, this is a basic of science, that if you are going to heat something, then the particles are going to be thin, going to move apart. Likewise, if the temperature is not favorable, then many of the microbes are not going to survive. And only those microbes are going to survive which are capable of tolerating this harshness of temperature both way, either very excessively high temperature or cold temperature. So the hypothermic and hyperthermic, both the conditions are not good. Relative humidity and moisture. How about that? Moisture is life, meaning water is life. And there is a lot of school beliefs that mold is the reason for moisture. If you don't have the moisture, you are not vulnerable to have the mold growth. So if your temperature, your humidity is in the combination of optical zone or optimal zone, then you have chances of getting mold proliferate, or bacteria grow, or virus can thrive. Other thing which I want to put a little bit of touch on that, because recently I start noticing that the light source also has impact. And I'm not the first person to observe that. A lot of other person observe it. And they have also come up, because if it is a damp environment, then the chances of microbial proliferation is greater than otherwise when you have light. Why? Because most of the microbes are pathogenic. They are heterotroph, meaning they have to depend on someone for the food, not on McDonald's or Chick-fil-A, because they don't eat that kind of food. But they need to have some organic material as a food. And many of you notice that if you go in a dark and humid and a place where the light is not that intense, what we call the perfect place or the ideal place for the microbial growth. And then sound also has impact. If you are continuously in a very noisy area, say decibel, in terms of decibel, that's the unit which you measure, use in measuring the sound. And if you are more than 80 decibel for a continuous or prolonged period, maybe like 8 to 10 hours continuously exposed to that sound level, it is annoying. That is also a type of contaminant. More or less, it also impacts.
So once we understand the types of contaminants that we can come up against inside of a building, and this sort of transitions us now into getting into the study and why we're studying bipolar ionization, but it helps us understand how to maintain or enhance, as you said earlier, Dr. Sahay, acceptable indoor environmental or indoor air quality. And so these 5 bullet points are sort of the main points that we think about when we start to develop a plan to either maintain or optimize the indoor environment.
Correct. And as you can see, this is one of the very important component of the bigger equation. Most of the built environment is designed based on its function, its type, which we have alluded earlier. But this is the lungs of the building. So if your lungs is not good, how good is that to support your whole physiology? Because lungs is the place where your oxygen exchange happens. Likewise, the entire building is getting fed the air through these units. As I said you that it is taken over by the mechanical system. So if the mechanical system is itself ill or itself not doing good, what do you expect from it? For an example, this picture, it's a graphic illustration, and Troy did a very good job to put this picture here. That suppose this is the filtration area, and this is your like coil, if you understand that, and it is malfunctioning, then all these debris which is coming to this unit is going to be just start blocking it. Not only blocking, because obviously the biofilm is going to form, or other thin layer is going to form. So it is going to impact in 2 ways. Number one, your energy cost is going to be much more, because the whole sum of the airflow is going to have a restriction. So you require more force to push it through. And if you require more force, that is directly the more energy consumption. Number two, that these things provide an ideal situation or perfect condition for microbe also to proliferate. So the proliferation of microbe happen, and can you believe that bizarre or a very huge quantity of microbes are going to push through these units and going in your livable space, and that's what you're going to breathe. And of course, no brainer, that is going to have some toll on your body or occupant's body. So see, in an ideal situation, look in the second diagram. You do not see any kind of debris coming in, first of all. So you are starting with a good note. So you need to be maintaining the equipment. That's why I said adequate equipment and adequate operation and maintenance. You have adequate equipment, but it is not maintained properly. How good is that? And now, many times people ask me, so what do we do? How you find out that whether it is adequately maintained or not? Then you need to do some testing and diagnostics on it. Time to time, on periodical basis, either you can, as simple as you can do look, see, smell, like, 3 basic things. And if you have some kind of, compromise there, then you have to do some laboratory testing in order to find out that what is going on. And then that will lead you, and I underscore, if you do these tasks, then only it will lead you to practice evidence-based management. Otherwise, it is just guessing game. I can be here guessing all day long that, hey, Troy, this is bacteria, this is fungus. But unless and until I have an evidence, because in science you have to have some kind of evidence to identify that, yes, it is a potentially a bacterial source or it is having a fungal source. And then I will be more in position to advise. So in nutshell, that where the evidence utilized for doing the operation and maintenance in a way that it's supposed to be done.
Yeah, I'll also mention, Dr. Sahay, that much of this is covered in ASHRAE 62.1, the 62.1 standard, especially in Chapter 8, where it revolves around creating an operations and maintenance manual for your HVAC equipment that includes just what you said, periodic testing and diagnostics on this equipment to make sure it's operating as it was designed and that it's adequate for the space that it's servicing. So once you have that information in hand from your testing and diagnostic studies, now we can create a plan for how to actually manage the indoor environment. Is that right, Dr. Sahay? And I will say, Dr. Sahay put this flow chart together because there's so many of them out there. And we know our audience is pretty diverse, but this is a very simplified version of how this kind of management system should be conducted.
Exactly, because I try to simplify because I do not want to use a lot of jargon. So I want to just give a very good idea that where to begin. So the total maintenance and optimization process or procedure should be depending on few very critical steps. What are those steps? So you do ABC of building. So first start with the building design. Whether the building is designed adequate for the occupancy that it is intended to, or it is happening. That is the first thing. Now, you cannot run a hospital out of a industrial building because it's going to be miserably fail because the open space or warehouses are not designed for running a hospital. So when you have to do, then what you have to do? You have to do certain engineering change if you want to do that. So I will talk about that a little later. So the building design should be evaluated based on the utilization, based on how much air need is there, and whether it is adequately maintained or not. Make sense? Now, diagnostics, like if you are having some fall, like I said, you look, see, a smell. If you are thinking something is not right, then what you got in your hand, that is called as diagnostics. So it can be proactive, or it can be reactive, or it can be monitoring. Proactive is a good approach because you know that what the building condition otherwise is. So when some complaint or some complainant come to you, then you can also compare those complaints against the proactively collected data. So that will help you to design the perfect management in that kind of scenario. Or you can have another approach that nowadays technology has gone up. So you have tool in your hand that call as monitoring. So you can monitor the condition which can supportive or can be preventive for the contaminant in the building. So there is 3 steps. There you go. And then occupancy in the building. whether the same people are working or living in that building each day and every day, or somebody is temporarily visiting the building, maybe staying 2 days, 3 days, 5 days office, they're coming and they're off. And there is a transit, because there are some people come for an hour, 2 hours a day. Those are the visitors. And why I want to pay attention there, that each individual may carry some contaminant, and don't laugh at me that they don't, because if they are working or going in the building, approaching in the building, and if they are walking on the field, as simple as that, their shoes and their other clothing and things like that, bringing the contaminant which is floating outside, and that is going to be set there. So these three approach is a good initial approach to think about the maintenance and optimization. And what you do after these three stages, you are going to have a risk assessment. So that will help you or lead you to determine the source, whether the building itself is a source or it is the visitor they bring those kind of source. So then you are going to understand that these sources are biological in origin, that is biogenic, or abiological in origin, abiogenic, or none of it. It may be the environmental condition. Somebody come with some kind of gas or something, and without knowing or knowing, that person releases some gas there. So that becomes neither biological nor abiological, but temperature raise, humidity raise due to that gas leak can happen in the building. So that can be contribution to the environmental factor. And once you have that idea, then you can do two approach. Routinely you do something as an intervention, so that even though they are bringing, you are checking, balancing that, so it is not going to create further problem down the line. Or you approach in a way that you are targeting that particular risk. For an example, if you are having a risk of COVID-19 for coronavirus infection, then what you're going to do? You're going to approach in a way that you are going to target SARS-CoV-2 virus that create COVID-19 infection or disease. So that is a targeted approach. So how to do that? Those approach can be handled either utilizing air cleaning technology or engineering intervention. I was telling you engineering intervention that you can check your HVAC unit or air conveyance system and make a due modification, or make a partition, or make a specify that particular area in the building for a special function, so that you can make those kind of engineering changes. But in the air cleaning technology, you can use filtration, you can use UVC, you can use ionization, and you can use like decontamination by utilizing various kind of product available in the market. So our goal today is to share some of the information that we have gathered by doing the experimentation on a technology or air cleaning technology that is called as ionization. So we have a study that whether the bipolar ionization has any impact on contaminant or not. So real quickly, I want to explain, those who are not aware, I'm pretty sure many of you are aware, that what is bipolar ionization. Bipolar ionization is a process in which negative and positive ions are generated from air, gas, or oxygen discharging electric or electric volt. So it's a plasma state. Matter has three states. Everybody knows, like solid, liquid, and gas. But there is another stage called as plasma stage, where the ionization of the element happen and they are existing in ionic form. So that is negative and positively charged ion, and that is the way it is being generated, and that is called as ionization or bipolar ionization. So what happened in that? As you can see here, that the oxygen, and this is the molecule, and then electron capture. So either they gain one electron or lose electron, and then they make this kind of complex agglomerated or conglomerated form. So what happened? The negative or positively ions collide with aerosol particles, which is existing, like what is existing in the building, both abiological or biological, to bind them together in a cluster large enough that can be filtered or captured or settled by the gravity. That makes sense? So that was the thought behind, and they thought that if this is going to be captured or this is going to be going against the gravity and going to settle, then the problem is going to be solved, because those contaminants are not going to be floating around, and subsequently it is going to be eliminated or at least managed, so you are not going to get the exposure out of it.
Yeah, that makes sense. And again, that's the theory of how it works, and not only in the building, but inside of the air conveyance system, where typically this technology is mounted or installed. Is that right, Dr. Sahay?
Yes, and that's that's what we are going to discuss in our study, because it really where you mount, how much ion is produced, all those kind of answers we are going to look. So I'm glad that Troy has put this together, and you can see this is a paper published by Kaba. And what we did in that paper, we evaluate bipolar ionization technology in controlling the hazard in indoor environment or indoor sites. So our goal was to find out that whether this technology has any impact on the microbiological entities, any impact on the chemical entities, such as chemical substances such as volatile organic compound, etc., or any other thing including the environmental condition, it has an impact. And how we can use this technology in maintaining a smart building. Those who are not aware, I don't want to, because this webinar is not about the smart building, but a smart building is those building which is typically referred that works on a set forth parameter, so that you can avoid various problems, because their temperature, their humidity, their airflow, etc. is being set up. So what we did in the laboratory experimentation, we have taken some devices, randomly selected devices, and we put them in a blower case assembly, and that box was, I put the size here, 28.5 inches, 15 inches, and placed in front of a fan. That fan was capable of moving 1200 CFM of air, so that the generated ion will be split throughout the exhaust of the blower assembly box, and there was an opening in that box which was inside this test chamber. So it is not SUBOS experiment. It is a full-blown environmental test chamber which is mimicking an office building, 10 by 10 by 8. That is the dimension of this test chamber. That's why I put the photo here so people can understand. And this is the blower assembly which I'm talking about, is running the air. But this thing, the blower assembly where we put the bipolar ionization, is sit inside this chamber. So what we did afterward, and of course it was connected with a 110 volt supply, and then we did our experiment. Can you go in the next slide, please? So very first thing, we evaluated the device which we have considered for this experiment, and it was totally randomized. It was not influenced by one vendor or the other, so I do not want to mention a name of anybody, but this is simply the technology testified that it is a BPI, meaning bipolar ionizer. So what we did, we mentioned that how much ions are being discharged, and this table represent. So you can see that we have a baseline, we have a device off, and we have a device on. We did purposely to figure it out that what is the natural existence of the bipolar ions, meaning negative ion or positive ion, because these are naturally occurring thing in the environment. And then when we move a device, then our goal was to find out that if any kind of device is going to go there, maybe there is some magnet or something else, whether it interfere in the natural existence or not. And the third goal was to find out that if we put the device on, how much ion we are going to get. And that number is presented in this table. And in nutshell, you can see that 92.20, meaning like just say like 92 million ions were produced, because 92.2 times 10 raised to the power 3, that's the number of ion we produce. So that was ion in terms of negative ion was produced from this machine or this device, and in terms of positive, it was about 59, just say 60. Okay, next slide, please. Our next goal, and a lot of people ask me a question that if it is that producing the ion, that how good is good? How much ion supposed to be there to kill? And that was our experimental goal to find out. So I showed you graphically that this is when the equipment is off, and this is the number when equipment is on in terms of like a negative. The red bar represent the negative ions, and the blue bar represent the positive ion. But at the same time, a lot of people ask me a question that whether this produce ozone or not. Because if it is producing a lot of ozone, then that is also not good. So I evaluated to find out that how much ozone it produces, and I was thinking 0.05 is a threshold cutoff for the ozone. So my goal was to determine whether it is going in that zone or not, this particular BPI. And I presented that data right here, which is not crossing more than 0.01, or it is definitely not reaching 0.012 ppm. So ozone production wise, this particular test device or the BPI device passes, because this can be used unless and until zero ozone production. If you are looking for the zero ozone production, this particular device is not fit for that. Next one, please. Now, the second thing is, and it's a very meat of this presentation, the core of this presentation, our goal was to find out what kind of impact it has on microbiological entity. So what we did, we have selected a bacteria called as Staphylococcus aureus, which is a typically or commonly reported from indoor environment. And this is a unique organism because it can be poised, or it can be in between, like, you know, the virus and the microbes. On the one hand, it is tough enough so that you can compare it with the mold. On the other hand, it is small enough to be compared with the viral particle. So that's why I'm just saying that why we chose that challenge. But irrespective of that, you can see in my table that we have done the experiment, and we find that there was 99.87% reduction of bacteria happen when the bipolar ionizer under the given test condition, which I said you, expose 180 minutes. That is the time of exposure when the data was collected. So it has some impact on the microbe, and that is the data, and that's what I want to share with you. And a pictorial evidence is here. This is like, you know, we have conducted this experiment for two sets, set 1 and set B, And you can see the data is not very clear here. Can we make a little bit bigger? Yes, we can. So if you can zoom in, you can see that like there is a lot of bacteria and subsequently it is going to go down. So 180 is going to be a lot less bacteria than 0 minute. That's what tells me the reduction. Next one, please.
Okay.
Okay. Now, how about the other thing? Temperature, humidity, carbon dioxide, volatile organic compound, and PM, particle matters. That is another few very commonly encountered contaminant in the indoor environment. Well, my result of this experiment is not very consistent, first of all, on these parameters. Because as you can see yourself, that if I go temperature, like here when we were running, so I won't be saying a very good impact on any kind of temperature because it begin with 82 degree Fahrenheit, end up 83. So 0.1 degree Fahrenheit is not like, telling me a greater reduction or benefit or any other kind of, conclusion I can draw, because this conclusion not fair to draw because, it's 82, 82 and then 83, 83. Now, if I go ahead and check the relative humidity, what happened? There is a 4% drop in the humidity. But the problem is this, the data is sporadic. First it's 54, gone to 51, 52, and 50. So yes, definitely it is having some kind of trend, getting the RH low, but again, this is not something which I can consider as a wow factor, that all of a sudden humidity is controlled in such way. Now, how about the carbon dioxide? Again, there is a trend here. Carbon dioxide is also going in the downtrend. The most interesting result is in terms of volatile organic compound. And here I want to explain that what I can gather with this. As you can see, this data is also very sporadic because we started with over 11,000 and all of a sudden it come to 2,000 or over 2,000 and then 1,000 and then gone to 10,000 and then again back to 2,000. So what it is telling me that the volatile organic compound may be first splitted or depleted, so the number go all of a sudden down, but they may have a chance of regroup again as a terms of byproduct, and then they come as a byproduct, but again this system kick in and start depleting that byproduct. But I do not have enough evidence to identify that what kind of byproduct was made and how it was depleted. I do not have enough data for that, and it required a little bit more investigation. But if somebody do the research, and I know that some of the literature out there that people have identified type of chemicals, and then you can figure it out that what happened, what is all the regrouping is about, and how it is being depleted. So if you see the result, the net result is telling me that it can be also useful for controlling the VOC, but I do not have any evidence that what happened in between because I didn't identify any chemical or their byproducts. All I identified the volatile organic compound in terms of their totality.
Yeah.
Then you have the particulate matters like 2.5. It's a very fine particle and it can go in your lower alveoli. And definitely this has shown me reduction. So I think the theory which was given that it is conglomerates due to the ionization or charging of the molecule. So either it is going to be filtered or it is going to be die down against the gravity. And that result is indicating that because as you can see, we started with 21 and then next is 25 and then 0, 0, 0, meaning it is getting either filtered out or settled that small particles. And same thing, I can tell you that the particle matter 10, which is a little heavier particle or little bigger particle, has almost shown the same kind of trend. So it might work on that. Next one. And this is simply the graphic representation which I just explained you. As you can see, the blue is one which is like machine is off, and this color like pinkish color or brownish color is representing that when the machine is on. So if the machine is on, see there is a trend, like it is getting like, you know, down. But when the machine is not on, it is almost at the same level. Except this is like, you know, the carbon dioxide, as I said, you a little bit of degradation. Such as volatile organic compounds, see that it is also telling like, you know, when the machine was on this is a big chart, gone down, down further, come up, and again gone very low. So that's why I was telling you the regrouping theory, like maybe they have byproduct again work, and then that might be, but again, I didn't identify any chemical or anything. Otherwise, I would have been in a better position to tell you that exactly what is the byproduct of the chemical and how that works. And this is the PM, which is obvious. As you can see, I told you that in these hours of exposure, there was no particulate matter reported. So definitely it has screened out. Next one, please.
So this was the evidence, finding that you were able to conclude from the data. So what were the conclusions you were able to draw from this study?
Okay, the real quickly, I'm going to touch the conclusions because I guess we do not have a lot of time here. So my experiment during these findings, our negative ion production was about 60 million, and the positive ion, just say give and take, 92 million. That was the production of the ions we have recorded from that particular instrument or the device. Then it is also saying that the indoor microbes and particulate matters were like, you know, affected with this, meaning that it has some impact on those indoor microbes and particulate matter. The tested microbe was Staphylococcus aureus, but the efficacy on the VOC and CO2 was there, but it was very sporadic because there was no clear-cut evidence that it is going to do that, because as I said you, there was little up and down. And the finding suggests that these kind of thing can be not solely depend to manage the indoor environment. Rather, it has a good addition to make your air cleaning technology or air cleaning monitoring or management in your place with this unit, because that will be help. This is not going to be just like, you know, solve your problem, because if you have a cancer and you are trying to treat it with the aspirin, not going to go away, because it might be pain will be down for a minute, but again it will come. So based on that logic, that the devices really produce or not produce, how much it need to be there, so a lot of things need to be considered. So the optimization of IAQ is not only the factor of the bipolar ionization, but it has number of other factor built in order to optimize your IAQ. So BPI is not the 100% total solution, but it is definitely a good addition to your fleet of managing or optimizing IAQ.
Well, right. You have to think of it like this, Dr. Sahay and viewers, is that no single area technology, filtration alone isn't good. UV alone is not good. Rather a balanced approach where everything is maintained, you're keeping an eye on it, and having all of these things work together is what really forms a picture of good or acceptable IAQ. So there's no silver bullet. We can't view this technology as the single thing that's going to solve all of our problems during COVID-19 or even after, right? There's no silver bullet, but you have to take that balanced approach. So Dr. Sahay, we do have lots of questions as we're wrapping up today. This is just a little bit about ED Lab. Obviously, your focus, Dr. Sahay, is on microbiology with a specialty in aerobiology and actually mycology as well, the study of mold. and you look at all of these things that we talked about at the beginning of the presentation, all of the indoor contaminants, and you're able to actually test for SARS-CoV-2 from surfaces and bioaerosol samples. And then these are just some of your accreditations, the big one being, of course, A2LA has accredited you with a certificate. And of course, the Environmental Diagnostics Laboratory is part of the CDC Elite program for Legionella testing. And so about Pure Air Control Services, well, you know, we've been founded since 1984. So indoor air quality is kind of our thing. We've been at it a long time, way before this pandemic started, and we're not product centric. We are basically service centric, and then we can recommend products that fit into that bigger picture, like you noticed there. So we have 3 specialized divisions, and that's our building sciences division that goes out into the field and collects samples and tests the performance of buildings, both from an environmental standpoint, but also from a design standpoint. And they bring those samples back to Dr. Sahay and his team, and they're able to study them in his laboratory. And then based on those findings, based on the evidence presented, we're able to go out into buildings and make suggestions for solutions on how to remediate any deficiencies that we find, whether it's in the air conveyance system, it's in the occupiable space, and we're able to sort of take care of that. All told, we have over 800 million square feet of experience and over 15,000 buildings studied worldwide. And so we operate in just about any type of commercial sort of non-residential building. And of course, all of our teams go through the rigorous testing and certification processes relative to their areas of expertise. So Doc, let's jump right into some questions here, because I know I want to be respectful of everyone's time. And so let's see here. We have a question from, okay, so when you were talking about the positive and negative ions, Alejandro Rodas has asked that what types of ions are they? He says that he believes that there are 7 oxidants that can form from an ionization process. So for this study, what were the positive and negative ions?
We haven't categorized them. We have categorized only either negatively charged ion or positively charged ion. So our equipment was not categorizing them, rather it was a broader group like negatively charged ion or positively charged ion. So 2 categories.
I gotcha. And I think that this question from Paul Haas was answered, but he asked, were different relative humidity ranges tested?
Yes, the humidity range tested for this particular study was between 58% to 62%, or sometime plus minus 1% variation.
And so he was also asking, is the corollary between the relative humidity and ozone concentrations, was it negatively or positively associated during the duration when it was on? So I think we sort of looked at that in the slide, right? This one where we looked at the humidity sort of went down and then up again.
That's what I said, like, you know, the 56%, and that's what I make a comment, that 56% to 57% plus minus 1% I said. So yep. This is the data which I presented here. So the humidity was like, you know, during the test, it is baseline was 56%, 56 point something. So that's why I said 57% give and take, but 56% is the rough number. And the 50.
The other part of that question was about the ozone concentrations. And I think that during this part of the presentation, you said that it never got above 0.05, right? Which is
0.012. This never gone above 0.012, because see on the scale, it is like 0.012. So none of the bar is touching 0.012. So this is like, unless and until your requirement is to have ozone production 0, then this particular device is not going to be work, because it is going to give you some ozone, but it is not going to give you enough ozone that you have the health concern.
Yeah, that's correct. And I believe that like California, the state of California has some of the most stringent regulations against ozone. And I believe, isn't that 0.05 is their tolerance for ozone? So this
Yes,
was still well below that.
yeah, there is two, like one is 0.05, other is 0.005, and then other one is a no ozone production.
Okay, and then we had another question here from Alan Messer. And Alan asked, the colony reduction appeared to be 98% with the ionizer off. He asked if you could maybe explain what the scenario was there.
Okay, so when the ionizer off, the colony reduction was 98%. Okay, and when the ionization on, then it was 99.87%. So if you can see in this figure, in nutshell, that what happened, that like when the ion was off, it was about above 14,000, over 14,000 colony. We left with only 18 colony. When we didn't do the ionic treatment, then as you can see that total ion was again for over 14,000 colony forming unit, and we end up with 284 colony forming unit. So definitely the 18 colony forming unit in terms of 284 colony forming unit is much less, and that's why it is 99.87% reduction. So it has some impact on the colony forming, so there was a reduction. Make sense?
It does. Well, to me, yes, it makes sense. It looks like it didn't do much more than having it off. If you just look at the percentile, but if you look at the actual numbers of colony forming units, then you do see that there was some change for the better.
Yes,
with the ionizer
because
on.
the thing is here, Troy, one more thing I want to tell Alan, that one colony forming unit can be trillions and trillions of bacterial cells. So it very quickly multiply. So even you are reducing one colony, you are eliminating the risk of, like, several million bacteria, because one colony forming unit is just not one bacteria. It is the summation of, like, as I said, you millions and trillions of bacterial cells, then you can see one colony forming unit. So having said that, if you go on that logic, then you can see that we end up 284 colony forming unit if you do not have that unit, this bipolar ionization. And if you use the bipolar ionization, it is 18. And that's why I make a statement that this is not a silver bullet. You do not consider that you are just going to put this unit and it is going to clean all your indoor air quality problem, all the microbes are gone. That's not happening. So what it is useful for, that if you are properly maintaining your unit, your air conveyance system, and if you put this kind of, like, device there, that is going to support you to optimize or maintain your quality of air or quality of environment as such.
I understand. We have Matt Davidson. Davidson has asked if we tested for UL 2998, or said, did you guys test a UL 2998 needle point ionization device along with the other random bipolar devices? I believe the device that we tested was UL 2998 listed, right?
Yes, it is. Like, the thing is that our goal was not that specific, so we may be doing in the future, but at this time we have done a lot of, devices, and due to the customer, like, confidentiality, I cannot tell the name of everything, but we have tested almost, like, all representative devices of, like, bipolar ionization. Some of them which we have published the data, many other data which we have collected is not published. And with that, having said that, I want to make an observation here that we have not only, like, studied this thing to see the efficacy, but we also study the thing that how many ion it produce and what is the impact of the ion in terms of, like, traveling, in terms of the time intensity, in terms of, like, their total concentration at a given point of time. So yes, we have done some experiment, but we are not very specific on the devices. No.
I got you. And then Patrick Meckelberg has also asked about the VOCs and such. He said carbon is the typical solution for VOCs, I guess meaning filtration, right? Under what
Yes.
situations would this need supplementing or swapping out with bipolar ionization?
Well, this is a very excellent question, an intelligent question. Like I was telling you that in this particular study, I haven't identified specifically the volatile organic compound. Rather, I have used to monitor the total concentration of volatile organic compound. So what this thing going to be useful in the real world scenario, that remember I was telling a logic agglomerated and conglomerated. If your volatile organic compound is floating in the air, then this bipolar ionization is going to deplete it. So when it depletes it, it becomes charged molecule. So when it is a charged molecule, then it is going to regroup them. Perhaps that's why my data is saying that the volatile organic compound is going up in 120 minutes, and then afterward that goes down 2000. But when I started, there was over 11,000 particle of the volatile organic compound. So that indicates that, like, if your system is otherwise clean, if there is a volatile organic compound, this technology will work to deplete it, make it like ionizing form, and then that will be regroup it. And then if it is going to go through the filter, it is easy on filter to absorb or what you call is scrutinize there, or take out from the air stream. So in that way, this will be having some usefulness in the scenario which you just asked me.
Yeah, and as part of the overall conversation that I'm seeing within the chat where the questions are being answered or asked, Paul Haas says that that's sort of where gas phase filtration would come in, which is part of ASHRAE 92. And I would imagine that would be used if you're up against a large volume of VOCs, like maybe a paint factory or a chemical factory, or compounding laboratories, or anything that would be producing a large amount of VOCs. For most cases, I would imagine, we're sort of studying this as if you were in a school or an office where there might be normal off-gassing of things like furniture or carpet, or even cleaning supplies, which of course that has been a topic during this pandemic, over cleaning by housekeeping, where they're using lots and lots of extra cleaning solutions that might produce many more VOCs than the occupants are used to. But certainly that's a good observation,
Yes.
as the gas phase filtration, would obviously probably take care of most common VOCs. Is that right, Dr. Sahay?
Yes, it will, and you make a very good assumption here, sort of like, your opinion that this particular device we have taken under consideration for our experimental procedure to see that whether it fits in indoor air quality scenario in our day-to-day life. It is not like meant for cleaning the off gases or any kind of gaseous volatile organic compound on a big scale, because there is no enough evidence that it is going to suggest that it is going to take out the VOC in a big compounding factory or big places, big industry where we are dealing with, like, large volume of volatile organic compound. So that's a true statement.
And then Mr. Haas did want to clarify, since we are recording this for posterity, that he meant ASHRAE 52-1 1992. I think that's where he got confused, and the technical fact ID 92. So for other listeners, viewers out there, that would be what he was referencing with the gas phase filtration. So I don't think we have any more questions. These were some really hard-hitting, good questions around this study. I do think that part of the overall conclusions are that more in-depth studies would need to be done, specifically in areas of focus such as VOCs, such as, like you said, Dr. Sahay, the distance that these ions could travel in any sort of volume before they need to be boosted again. We know a lot of in-room air purification units that have their own fans. Some of them have bipolar, needlepoint bipolar ionization that could maybe supplement the occupiable space if the unit that's inside of the air conveyance system is far away from the rooms where occupants might be. So that might be helpful, but again, more studies would be needed that would have that specific construct. Is that right?
Yes, we have also done some other studies which is not published as yet, but like you were saying that we have also done a study that what is the fate of the ions, like, traveling in the duct, like, how it is viable, non-viable, getting lost. These kind of a study we have done, like, at the source from where it is produced, being traveling up to 25 foot in the duct. Then what kind of, like, data we get, we have gathered those data, but we haven't published those papers as yet. Maybe we can take some other webinar to explain the several other variants, because there are a lot of variants here. One of the good variants, somebody asked about the humidity. Still, I'm interested knowing the more about, like, if the humidity go, like, 55% or less, or like it goes, like, over 60%. What happened? That I really want to know. I'm dying for that, and my goal and focus is that. That's why I said, in general, like, my belief with my experiment is, like, typically the 58 to 62% humidity, this should be stand valid, the result which I have produced today, because quite frankly I do not believe that it is going to disturb between 58 to 62%, whereas my experiment in the test chamber we have maintained, like as I showed you, like, 56% to 50% we have recorded that on our monitoring. So there are a lot of variants there. We need to definitely understand this device more. But as a concluding remarks, I think that it is a good supplemental factor for indoor air quality purposes in combination of other air cleaning technology. And of course, like, the maintenance is the key element, because if your system is not maintained or your duct is not, like, maintained or optimized, do not, like, think that this is going to create a magic wand and going to create solve all the created problem, because many time I have noticed, just talking to our customers, that first thing to go, they don't pay attention about the maintenance if the beds are tight, any organization pretty much. So that comes, like, that is not getting that much of priority, and that is one of the concerns that worries me. And other than that, like, it would be a good substantial like, factor to tell someone that if this is going to have in their system, this is going to help to robust or boost at least the air purification, the whole process, but not alone. It is not going to solve the problem.
No. So, well, folks, we're up against our hour. We certainly thank all of you for hanging in there with us and the wonderful in-depth questions that you asked. As you can see, here's our contact information. So feel free to call or email us anytime if you think of any more questions, or you want to take a deep dive into some of the subject matter. We're happy to talk to you or put you in touch with the right person here to talk about it. And of course, there is a survey upon exiting the webinar. We would appreciate your feedback on this topic. And of course, like I said, don't forget there's handouts. So this slide deck is in there, as well as the white paper itself, and then a little bit of background information on the environmental test chamber. So the video replay of this will be sent to all of you, so you can share with your colleagues. And of course, it will end up living on the Pure Air Control Services YouTube channel. So on behalf of myself and Dr. Sahay, we wish you healthy buildings and healthy people.